So now we're going to do part 2 of chapter 18. We are continuing on with our discussion on bacterial pathogens, and then we'll look at parasites and viruses, and then just some closing things, including food safety prevention, or not prevention of food safety, but food safety preventative measures that the industry uses, and you'll want to pay attention on that because that will have something to do with your product development projects that start this week. So let's move forward. Okay, so foodborne infection, we covered foodborne intoxication last week, and we're going to do foodborne infection bacteria this week. And in the case of foodborne infection, these microbes, they enter the body, and once they are in your body, they release the digestive enzymes that damage body tissue. So there's no toxins released, it is just enzymes that eat away at your body tissues because that's what they do. They are made for digestion, and that causes the illness. So these foodborne infections, they may be caused by bacteria, parasites, or viruses. And in general, there is a large number of organisms that is required to cause illness. Now, in some cases, you must consume a large number of organisms, but also in many cases, that large number of organisms accumulates in your body and then causes illness. So it's not necessarily the case that you have to consume a large number. So the first one we'll cover is Listeria monocytogenes. For this one, only about 1,600 cases per year are reported. Of that, it's a substantial number that die. I mean, more than 10%, 260 deaths per year. And we'll get to why it's really a big focus. So the symptoms of that is that it causes fever, headache, and then our general gastroenteritis symptoms, nausea, diarrhea, and vomiting. What really comes into play with Listeria is that it can cause miscarriage or stillbirth. So it's a doozy for pregnant women if they contract it. 25% of the serious cases result in death. And so this is substantially higher than a lot of these other foodborne infections. So we don't have a lot of these foodborne infections per year. 1,600 cases being reported is not really a lot, and based on the size of the United States and compared to other foodborne pathogens, but it can be really serious. And so there's a lot of attention in the food industry paid towards Listeria monocytogenes. It very easily can get in food. It is ubiquitous in the environment, like a lot of these foodborne pathogens. It is actually psychotrophic, and this is one of the things that makes it a serious problem. So psychotrophic means that it tolerates cold temperatures quite well and can even grow at cold temperatures. So pretty much all of our other microorganisms, as long as you keep the food at refrigeration temperatures, you are preventing growth of those microorganisms. They are mesophilic, they are purely mesophilic, and at refrigeration temperatures, they do not grow. So temperatures under 40, right? 40 to 140 is our danger zone, and anything under that, we consider that a good safe temperature to hold our food. But Listeria is a problem because if there is just one Listeria cell in that food, it can multiply at refrigeration temperatures and get to a point where there are high enough numbers to cause serious illness. So in foods that are going to be held at refrigeration temperatures and not cooked, there's a lot of attention paid towards Listeria monocytogenes. Anything that is considered ready to eat really has to have Listeria monocytogenes prevention measures. Listeria also tolerates salt quite well. So things like lunch meat, it's fine with. Salt. Salt is not a good preventer of Listeria monocytogenes growth. So, for example, when I worked in the meat industry, we made lots of processed meats products, and most of those are ready to eat. You think about hams and smoked sausages and lunch meat. So we had lots of preventive measures for Listeria monocytogenes. There were ingredients that were in the products that we required to be in all products that were considered ready to eat that are tested and we know prevent the growth of Listeria monocytogenes. These things are things like calcium lactate, for example. We also had tons of testing that would go on. Not necessarily on food products, but on all of our surfaces in the plants. We would be doing constant testing, and the goal, like we actually had a training program called Listeria Hunters, and the goal was to find it and find it in those itty bitty nooks and crannies before within the equipment and within the facility before it ever had a chance to escape those nooks and crannies and get onto food. So it's a really, really important issue in the food industry. And the thing to really know about Listeria monocytogenes is that it can grow at refrigeration temperatures and it causes miscarriages. So control of Listeria monocytogenes, it does not grow at pH 4.6. So if you have a ready to eat food that has a lower pH, you're pretty safe. It does die if you cook the food thoroughly. So that is why one of the recommendations for pregnant women that has been around for a long time is that if they are eating lunch meat or hot dogs, that they reheat those before they're cooked. Now that recommendation is a little bit outdated because like I said, there are ingredients in those foods that prevent Listeria growth today. So it's really not as necessary. As someone who's been pregnant before, I never worried about it, knowing what I know about how Listeria is controlled in those products. But if the products did not have those ingredients, yes, you would want to reheat those foods thoroughly to potentially get rid of any Listeria that was growing in them. I'll also say, so one of the things that we also did in the industry was we would have our own studies that, so with these ingredients that we were adding to the Listeria monocytogenes, we would have a batch of the product that had those ingredients in it, and we would inoculate those products with Listeria in the lab, right? It never goes into human consumption. And so we would actually add Listeria monocytogenes and measure the growth or death of the Listeria monocytogenes over time. And essentially what we wanted to see was a flat line, right? No growth whatsoever. And if there was any growth, then we knew that that ingredient was not a good controller of Listeria, and we either needed to increase its level or find a completely different ingredient to use. So it's a lot of testing that goes on behind the scenes to make sure that the food that is being produced is safe. Listeria can also, like I said, it's ubiquitous, so it can be found on raw vegetables. So wash those raw vegetables thoroughly. Sprouts, like I mentioned last week, as a potential carrier of E. coli, they can carry Listeria also. There have also been numerous outbreaks of Listeria in other produce. Cantaloupe is a big one. So just always be on the lookout for what produce is being recalled because things get contaminated and then there's massive outbreaks. There are actually probably more outbreaks of a lot of these foodborne pathogens in produce than in meat, even though meat is a better, uh, it's a better nutrient medium. It grows a lot of these pathogens better than produce, but we'll get into it at the end of this chapter. But there's so much, I would say regulation, but also just prevention going on in the meat industry, and a lot of the other parts of the food industry are behind compared to the meat industry, and you'll see why we're going to get there. So also avoid unpasteurized cheese, especially if you are susceptible, if you're pregnant. And especially if you are at risk, the recommendation, like I said, is to reheat pre-cooked foods. Personally, if you look at the ingredient statement, there are lactates and citrates and things like that that are in there that whose goal is to prevent listeria growth, and that recommendation isn't really that necessary anymore, at least for those products, for lunch meat and hot dogs. Okay, so salmonella is the next one on our list. That is an organism that causes a foodborne infection, not intoxication, infection. Okay, substantially more cases per year than listeria are reported for salmonella. So 1.35 million cases per year, 420 deaths. Now that number of deaths total is more than the listeria, but you notice that as a percentage wise, it's much, much lower. So generally, salmonella is a less severe illness, and it's really going to hurt those that have some kind of immunocompromisation. So salmonella causes diarrhea, cramps, fever, nausea, vomiting, chills, and headache. The timeline for this is that the symptoms start 6 to 48 hours after ingestion. So it can be as little as 6 hours or as much as 2 days. And the symptoms last for about 2 to 3 days. Now, I don't remember whether I mentioned this last week, but what you should have noticed by this point with the time after ingestion that you see illness, the symptoms start to occur, for the most part, with the exception of maybe Staph aureus, and even then it has a range, you cannot tell what it is that you got sick from just by the timing of it, okay? More than likely, when you start feeling sick, it is not the last thing that you ate, because the incubation for these things can be so long. So how do you find out what it was? Well, you know other people who ate the same thing at the same time. And that's really how they source those things out. If you end up in the hospital with this, they start doing an investigation. They find out all the things that you ate over the last 4 days. And when they find other people with the same symptoms and actually the same strain, they will do DNA tests to find if it was the same actual strain of bacteria that made you sick. DNA tests on the organisms. Then they start tracing back. Okay, so what is the commonality that all of these people ate, consumed, and that is how they trace it back. It is not a simple process. You can't judge what made you sick based on the last thing that you ate. The only thing that works faster is Staph aureus, and even then, sometimes the period before you get sick is multiple hours, and it might not be the last thing that you ate. So for salmonella, a large number is required for illness, but this one does definitely grow to a large number inside the intestine. So even if you consume a small amount of salmonella, it can grow in your intestines and then eventually make you ill. So, that's where this 48 hours comes into effect, right? If you could consume a large number, well, within 6 hours, you might get sick. But if it's having to grow quite a bit before it gets to large enough numbers in your body, then it's going to be closer to that 48-hour incubation period. Poultry and poultry products are the highest implicated. Of course, that one's probably easy for y'all, right? Chicken and eggs often are a concern for salmonella, and that is why we cook chicken to the temperature that we cook it, and why it's recommended that you don't consume raw eggs like in raw dough. But it is important to know that that's not the only source. Pork and beef can also contain salmonella. Raw produce, there have been lots of outbreaks of salmonella that have been due to raw produce. Flour can contain salmonella. And one of the tricky things about salmonella is that it can, when it dries out, it can actually be more resistant to being killed. So that's one of the things, like in the meat industry with jerky, we had to be very careful that we got things to a high enough temperature to kill salmonella before the meat got dried out, or we did other things to get rid of salmonella, because if there's salmonella on the surface and the meat is dried out and that salmonella is still alive, it can still cause issues. So how do we control salmonella? Keep cold foods cold and hot foods hot. And that prevents growth. It is purely mesophilic, so if it's at refrigeration temperatures, it won't grow. If it is at hot temperatures, it won't grow. But if it is in that danger zone, then it can grow. You prevent cross-contamination, right? Don't have your raw chicken and then and then on the same cutting board, cut your cooked chicken if you haven't washed it yet. Or don't cut your fruits and vegetables that you aren't going to cook on that raw cutting board. Cook ground meat and poultry until thoroughly done. So that means for ground beef and pork, that temperature that's recommended is 160. Now that is ground meat, okay? If it's whole intact muscle, you don't have to cook these to this temperature. You can cook it to whatever doneness you prefer. Poultry should always be cooked all the way through to 165 degrees Fahrenheit. And the book says make sure that the juices run clear. Do not rely on juices to determine whether your meat is done. Please, please, please. Use a meat thermometer. There are factors that can influence whether the juices run clear, and in some cases they might run clear and like just the color is changing of the meat juices at an earlier temperature than 165, but it hasn't actually reached that and it's not safe. Same goes for the opposite, all right? You can have pink meat and it has been cooked thoroughly. And I'm not going to go into the factors that affect that, but do not rely on the pinkness of meat or the juices. Okay, use a meat thermometer. And then finally, don't consume raw eggs or egg products unless they've been pasteurized. Okay, so that's all we're going to go through for bacterial foodborne infections. And now let's go into the foodborne parasitic infections. So what are parasites? Parasites live on and live and feed on the host plant or animal, and you do not want to be their next host. So they are found in potentially contaminated water. So examples of that are Giardia and Cryptosporidium, raw fish, and pigs and various wild animals. Okay, and these are pictures of all four of those. Just Giardia, Cryptosporidium, the Anisakis roundworms, and pigs and wild animals. I think we're only going to go more in depth with trichinosis, but just FYI, so contaminated water, that's an easy one. The raw fish, if it's in raw fish, they can be killed by freezing, and so there actually is a protocol for raw fish that are going to be consumed raw, because they'll be killed by cooking it too. But if they're going to be consumed raw, like for sushi, there's a protocol of freezing it first for a certain length of time, and that kills off the roundworm so that fish is safe. Okay, So Trichinella spiralis is the source of trichinosis, which is what it's called as an infection in the body. And it is a microscopic roundworm. So unlike, you know, this one, right, you have to use a microscope to actually see it. Uh, the host animals of these are pigs, bears, boars, rabbits, and potentially humans. It is actually in the ground in one of its stages. And so pigs, they root around a lot in the ground, and that is how they get that. Um, bears also digging things in the ground. Rabbits are very close to the ground and like eating roots and things. And then potentially humans because we're consuming one of these host animals. So the larvae, so you consume it, and then the worm will put cysts in your muscle, and it's painful. The symptoms of trichinosis. So when it's in the worm form in your digestive system, it can cause nausea, abdominal pain, and diarrhea. And then it lays those eggs, and the larvae get in your muscle, and that can cause muscle pain and fever. So how do we control Trichinella spiralis? So you cook the product to 160 degrees Fahrenheit. You can freeze the product for 20 days. So this is why maybe your moms, but definitely your grandmothers, have been always taught to cook pork very, very thoroughly, because pork historically was a source of this, and you had to make sure that you killed it. Today, it is actually not a problem. And that's because these animals are raised indoors with a very controlled feed. Everything that goes into their bodies is controlled. Versus, way back in the day when more often pigs were raised outside. So this is specific to commercially raised pork. If you're buying some pasture raised pork, then this is still a concern, and you need to make sure you're cooking your pork thoroughly. But today we can treat pork more like beef and choose whether you want it medium rare or well done. The recommended cooking temperature is minimum still 145 degrees Fahrenheit. So not rare for pork, but you can definitely cook pork at a lower temperature compared to 160 degrees Fahrenheit. And so there's a lot of history behind this, but a lot of people don't like pork, really myself included, to be honest, because it was always served to them like this, like a hockey puck, and it's dry and gross. It's also one of the reasons that pork almost always, when you buy it in the store, has been injected with a solution with sodium phosphate in it, and that helps keep it moist even if you do cook it to this really high temperature. And it adds a saltiness to it too. Uh, that's some of the history behind that. Little factoid, I got to be part of the research that determined this, not so much for the food safety standpoint, but for the sensory aspect of it when I was a master's student at A&M. So this came out while I was a master's student. All right, viral infections now. So, key thing to know, viruses do not multiply in food, but they can be transmitted through food. So they don't grow in food because they have to attach to a live cell to replicate. And there are no live cells that are in food that is already in its food form, right? So the host cell grows the virus particles, and then it ruptures and dies, is how viruses reproduce. Viruses can survive in adverse conditions for weeks, So they can survive at refrigeration temperatures. They can survive in dry environments. So they are not like bacteria in some ways. I mean, sometimes bacteria can survive these, but they don't grow, and the viruses are not going to grow at all. But they can survive in those conditions for weeks. So that is important to know. Viruses are destroyed when the food is cooked, no matter what. So most often, viral infections that specifically are foodborne. Okay, so not all viral infections follow this, but specifically if they're foodborne, they are most often going to be transmitted through some sort of fecal route. So you're getting it because someone didn't wash their hands after using the restroom, or perhaps there was water that was contaminated with sewage. More of an issue in non-developed countries than in the US, but still something to know. And or fertilizer on crops. So if people are using a manure fertilizer on crops, you risk spreading this virus. So there are 3 viral infections that we are going to focus on in this chapter. The first one is rotavirus. Most often this occurs in young children. It's pretty mild. It causes diarrhea, vomiting, low-grade fever. It's not common for it to cause severe issues that require hospitalization or death. But there is a vaccine for it. And after I just told you it's pretty mild, you kind of probably wonder, why is there a vaccine for it? And I wonder that too. Okay. Norovirus is probably the one that's most common that you've heard of. The first strain of this, the book calls this Norwalk virus. Okay, I'm calling it norovirus because Norwalk virus is actually a certain strain of norovirus and not encompassing all of them. But it's called norovirus after this first documented outbreak that occurred in Norwalk, Ohio, in some school children in 1968. Okay, there are 19 to 21 million cases annually. So this is by far the most common foodborne illness. There are 900 deaths and 109 hospitalizations annually. Most of these deaths and hospitalizations are in the very elderly that their bodies just can't take this kind of illness. In general, the symptoms are they have mild flu-like illness, so they can get headache and fever like the flu, but also gastroenteritis. And so this is the one that is most commonly, people are like, I had the stomach flu. Well, they had norovirus. They did not have the flu. They did not have influenza. They had norovirus. The best prevention for norovirus is good hygiene. Wash your hands, please. Okay, the third and final virus that we'll talk about is hepatitis A. This is a viral infection that attacks liver cells. The hepatitis means infection of the liver. Okay, hepatic refers to the liver. Hepatitis A is the only strain of hepatitis that is transmitted through contaminated food. Okay, there are other hepatitis strains, but those are not transmitted through food. For hepatitis A, there are 2,000 to 20,000 cases annually, so there's quite a bit of variation. Most recent year, there's closer to 2,000, but like 3 years prior, there were closer to 20,000. And you think, oh great, we're in a downward trend. Well, then you look a couple of years prior to that, and it was 2,000. So just annually, the number can vary quite a bit. So the transmission. One of the most common transmission causes is food handlers that are not practicing proper hygiene. And the problem with this is that it can be transmitted 7 to 10 days before symptoms develop. So they're not practicing proper hygiene, and they and no one else knows that they are sick at all. And then they're working in a restaurant. They are handling all the food, and lots of people get sick. And so hepatitis A, you quite often hear of outbreaks that are connected to a particular restaurant. It can also be transmitted from shellfish, and how shellfish get it is that they are in waters that have been contaminated with human sewage. So get yourself make sure your shellfish is from a reputable source. There is a vaccine for hepatitis A, and if you know that you have been, you generally don't get a vaccine just because. So what happens is there is a documented outbreak, and or there's an announcement, oh, we just found out the worker at this restaurant had hepatitis A. If you've eaten at this restaurant in the last 2 weeks, then please come in and we can give you this vaccine. And that can prevent you from getting the infection to a point where you exhibit any symptoms or can spread it to anyone else. Okay, so then the last thing that we'll talk about as far as foodborne infections is prions. So these are completely separate. These are not bacteria, parasites, or viruses, okay? But prions are actually a protein that is misfolded. So in this little image here, this is the correctly folded protein, and then something has happened, and it can happen actually naturally, but something has happened and it becomes misfolded and doesn't look the same anymore. Okay, and this protein cannot be broken down by the body. Okay, whether you consume it or it's already in your brain, and it aggregates in the brain tissue. And the thing that makes it really bad is that it is kind of like contagious within your body. This misfolded protein will cause others of these prion proteins to, this misfolded protein will cause other prion proteins to also become misfolded. And so it spreads in the area that it's at in your brain or your brain stem and accumulates and causes issues up to and including death. So in cattle, this is known as bovine spongiform encephalopathy when they have this. So BSE is the abbreviation, and the layman's term for this is mad cow disease. In sheep, it's known as scrapie, and it's called that because they actually will like scrape themselves along the rocks. It's part of like the mental illness that comes with the disease. And deer and elk can also get it, and it is called chronic wasting disease. Now, humans, if they get it, it is Creutzfeldt-Jakob disease. Creutzfeldt-Jakob is how it's pronounced. And in all of these forms, no matter which animal or human is getting it, it can happen spontaneously, usually in older animals. It's just things stop working as well and they break down and they don't fold right. There is a genetic component that more people are prone to getting Creutzfeldt-Jakob disease, or through eating meat with the prions in it. And so when that happens in humans, it is called variant Creutzfeldt-Jakob disease. Okay, variant. And so that shows that it is you've gotten it not from a natural spontaneous way that you've eaten the prions. So how is this spread? So cattle consume prions, and then we eat the meat that has the prions in it. Is how humans can get the variant Creutzfeldt-Jakob disease. And so some background to this, giving you a little history lesson. So in 1986, the United Kingdom began to notice this disease that was appearing in cattle, and they eventually traced it to feeding of products from scrapie infected sheep as well as meat and bone meal from cattle. And so they were feeding the cattle basically meat and bone meal from sheep and other cattle. And why in the world would they do this, right? Like cattle are not usually cannibals. But the reason that they did it was is that it actually improved milk yields. Okay, so most of the cattle, not all, but most of the cattle in the UK are dairy cattle. They have a big dairy industry, and so you're always trying to find ways to improve how much milk you're getting from the cows. And so they found that meat and bone meal improved the milk yield much better than plant sources of protein. And so that's why they were feeding these byproducts to cattle. Well, the problem is that dairy cows are not slaughtered until they are old, right? They have much longer lifespans than beef cattle. Beef cattle, they're slaughtered when they are like a year and a half old. Dairy cattle can live years as long as they are being productive. They are kept as dairy cattle, and then once they lose their productivity, then they move into slaughter and being used for beef. And then those byproducts would be dried, ground up, and then fed back to the cattle. So older animals are already more potentially prone to getting just the natural form of mad cow disease just because of their age. Like things just break down and happen. And so what happened was now these cattle were being fed meat and bone meal from older cattle that were more prone to getting this disease. They were contracting it, and the cycle just went up and up and up because now they had it, but you might not see those symptoms by the time that those cattle are slaughtered, or they might look like this. But then the products from that were still going into the meat and bone meal that was now fed back to cattle. And so you're feeding more and more and more contaminated product to these cattle over time as this cycle continues. So 1980s, late 1980s, they realized what was going on. And so the UK then started banning ruminant protein being fed back to ruminants. In the mid 1990s, they started banning the feeding of all mammalian meat and bone meal to animals, as well as its use in farm fertilizer. So that is a practice that has been used before too, is putting that meat and bone meal into the soil as a fertilizer. Then they also started testing all the cows that were over 30 months of age that were intended for human consumption to prevent it going into the food chain. But it was really too little too late. So 1995, the first 3 victims of variant Creutzfeldt-Jakob disease died in the UK. But eventually 178 people died from that in the United Kingdom. So while this is going on, everybody else in the world has banned beef from the United Kingdom. Over 180,000 cattle in the UK were found to be infected, and 4.4 million of them were 4.4 million total cattle were just slaughtered and disposed of, just burned in the fields in order to try to contain the disease. Like I said, beef from the UK was banned in most of the world. So huge impact on their economy and the farmers there. So continuing on, in 1997, there was a feed ban in the US and Canada. So we were like way late compared to the UK banning this type of feed being fed back to the animals. And we were just lucky that we didn't have the same issues that they did. But in 2003, There was a case in the U.S., the first case in the U.S. They called it the cow that stole Christmas because they found it on December 23rd of that year. And so like the industry was like, "Wah!" right before Christmas. And it was bad news. But it was a dairy cow from Canada that was born 4 months before the feed ban had been introduced in Canada. So, but after that, more than 30 countries banned U.S. beef, and it took more than 15 years for all these bans to be lifted. So it even had an impact on the U.S. And that was just based on one case, even though the cow came from Canada. So later that same year, just like a week later, the U.S. Banned any downer cattle from the food supply. So if a cattle can't stand up, they can't go into the food supply. And that's because this is a symptom of mad cow disease. As well as certain organs like the brain, spinal cord, and some other organisms that are known to potentially hold prions. Those are not allowed in the food supply either. So all in all, there have been 7 total cases of BSE in the U.S. Only the first case was this atypical form this form of BSE that is from consuming prions. The other 6 cases are an atypical form, which is the spontaneous form that just older cattle may just happen to get, and it's not considered to be actually contagious. So still, so the current measures to prevent this from happening in the U.S. It's illegal to feed cattle, sheep, goats, mammalian proteins that are derived from bone, blood, and organs, or any protein from an animal that has been infected. Like I said, there's been 7 total cases, and only one of them was a potentially contagious case that they found. And of course, it did not go into the food supply. They found it. Cooking does not, and there's also a lot of extra measures that are taken for any cattle that are over 30 months of age. So if they are over 2.5 years old, the carcasses are under increased scrutiny, and certain products are not allowed to be sold from those carcasses. So the key thing here with prions, cooking does not help. Okay, obviously, or so many people wouldn't have been sick. Also, downer cows are banned from the food supply, and how this works in your brain is that once you're infected, the prions that are in your brain contaminate other proteins, and it spreads within the brain. So if you are curious anymore about this, I put some videos on the YouTube channel. If you want to see what it looks like, what like a mad cow looks like, there's some videos of like live animals trying to walk and things, and they have a hard time walking. Okay, so that is the end of foodborne infections. And I know I went way more into that than you need to know, but I think it's just like an interesting thing to know exactly what mad cow disease is and how it's gone about and how we know we're safe now. Okay, so how pathogens enter the food supply. So one of the big problems with pathogens is that bacteria reproduce so quickly that new genetic strains can emerge in less than 24 hours, okay? So we can have full genetic changes in a matter of hours, not days, not years, but hours, okay? And these changes can be imparting antibiotic resistant. And so how that works is you have this group of bacteria, you apply an antibiotic, and you didn't kill all of them. A few of them survived. And those few that survived were the ones that were better at resisting the antibiotic. And they're going to reproduce and pass that resistance along. And so you end up with a new strain that is more resistant to that antibiotic that you applied. And so how that impacts the food industry is when animals are given antibiotics at low levels, and this has been the case that in some parts of like the livestock industry, they've given antibiotics at low levels to not to kill disease, but to actually improve performance of the animals, so the animals grow bigger, better, faster. Uh, and so they're giving it at low levels that aren't enough to actually kill all the bacteria. And so the bacteria that survive now become more and more resistant to that antibiotic, and you get antibiotic resistance. Okay. The same things can happen with temperature resistance. You didn't cook the product quite high enough to kill everything. So you have a few survive. Those few that survive are better at resisting temperature differences. And that could be heat or cold. Gradually, bacteria can be more resistant to cold, for example. You get strains that are resistant to cold. pH tolerance, air tolerance, all of these can potentially happen in populations of bacteria, and you can get new strains develop in, like I said, a matter of hours. So there are two ways that microbes get into the food supply. One is transmission by animals, and the other is improper food handling. So transmission by animals. So for many of these bacteria, it's transmitted through feces. So you have warm-blooded animal carriers, mammals or birds, and somehow the food is contaminated through the feces. So, example, E. coli in apples. How would an apple get E. coli? Well, deer have E. coli. They're walking amongst the trees. Workers are in the orchard picking the apples, and they are climbing up a tall ladder to get to the top of the tree. At some point, they are putting their hands on the same rungs that they previously stepped on, and the ground is littered with deer feces. Okay, so put your hands on the same rungs that you've stepped on with your shoes that have stepped in deer feces and grab that apple. You've now contaminated that apple with E. coli. Or the other situation that could occur with apples is that apples that fall on the ground before it's harvest time, well, those don't typically go to the grocery store. Right? Those are not the apples that you buy at the grocery store. But to utilize all of our resources and be efficient, those apples may often go into juice, right? They're too ugly, bruised, etc. to be sold whole, but they can still be used for juice. Well, they were on the ground though, and there is potentially E. coli on the ground. And so if you do not pasteurize that apple juice, you run the risk of it being contaminated and getting people sick. And that particular situation has happened before. We've also talked previously about how feces can get on the exterior of a carcass and then onto the exterior and then into the meat, particularly ground beef, where now the exterior becomes the interior because of the chopping and mixing. So other transmission by animals, raw fish. So I've already mentioned that there are parasites potentially in raw fish, and that source is going to be sushi or shellfish, and freezing kills those parasites. So that is a way to prevent that. Meat and dairy. So meat and dairy are, I'm not going to say the number one source of foodborne illness, because like I said before, there's so much preventive measure, but foodborne pathogens grow extremely well on meat and dairy, and they can potentially be a large source of foodborne illness if you're not taking proper precautions. So the 3 reasons for that, and you should know these. Meat and dairy, they provide a perfect medium in which microbes thrive. All right, the microbes that make us sick like to grow on in around bodies. Okay, that makes sense, right? If they can make us sick, then they prefer a biological medium that is like an animal to grow on. Okay, so the microbes thrive on it. Uh, and meat and dairy are popular foods. So the more a food is eaten, the more chances there are to make someone sick. Okay. And then finally, many people are uninformed about how to handle these foods. They think that, oh, when that chicken juice is pink, is pink, I shouldn't eat it, and if it's clear, I can. Or same thing for burgers. If the interior of that burger is not pink anymore, it's good to eat. Not the case. Uh, Use a thermometer. Uh, they aren't thinking about cross contamination and um, putting their cooked burgers on the same plate that they brought the raw burgers out to the grill with. So many people just don't have a full proper understanding of food safety and how to handle foods, keeping hot foods hot, cold foods cold, etc. So those are the three key reasons why meat and dairy products are particularly vulnerable, and a lot of food safety measures are put in place for those compared to other foods um, so that they don't infect people. So, so like I said, the first way that they get into the food supply is transmission by animals, and the second way is improper handling procedures. So, within improper handling procedures, we have time and temperature abuse. Okay, this is the number one cause of foodborne illness: the failure to properly cool food. Okay, so the book tells you there's a temperature danger zone. I have talked about a temperature danger zone several times, and I have not said the same temperature as the book because the book is wrong. I don't remember what the book says. Use this temperature, 40 to 140 degrees Fahrenheit. It's also easier to remember than the book temperature. Okay, 40 to 140 is the danger zone. Okay, no food should be in the danger zone for more than 2 hours. Okay, if your food is potentially going to be in that zone for more than 2 hours, you need to figure something else out to get that food cold or keep it hot. And this is whether it's before or after cooking. Okay, so don't set your meat on the counter to thaw or in a sink with cold water to thaw, because it will get into this danger zone. Even if it's not all of it, right? The middle is frozen, but the outside could still be in this danger zone. Not safe ways to handle your meat before cooking. Um, so keep your foods cold, hot foods hot, no more than 2 hours outside of that or within that danger zone. Okay, the second improper handling procedure that causes people to get sick is poor personal hygiene. Okay, this is both food handlers, so the people who are working in the food plants or the restaurants or I don't know, cafeterias, uh, but also consumers, right? Yourself, right? Make sure that when you're cooking food at home, you have good personal hygiene. So the ways that poor personal hygiene gets contaminants into food is, not washing your hands, breathing on food, accidentally getting hair into food. If you have an open wound, that is particularly a problem. Uh, Coughing and sneezing on food, perspiring on food, obvious things. Okay, and then the final improper handling procedure to be aware of is cross-contamination. So use separate cutting boards for your raw and cooked foods. It's even good to keep them separate, whether you're washing them in between or not. So like having a cutting board that only raw foods ever go on this cutting board, and having another one that only cooked foods ever come on that cutting board. And the reason why for that is when you are cutting into a cutting board, I'm sure you've seen older cutting boards, they have knife marks in them, right? These are little nooks and crannies that bacteria can hide in and resist being washed out of. And so you can actually still potentially get cross contamination even from a clean cutting board if it's used for raw foods. Changing plates between raw and cooked food. If you're taking something out to the grill, just get rid of that plate, get a new one for the cooked food. And be particularly careful when you have raw food that may be contaminated and food that won't be cooked at all. So, you know, if you have hamburger, raw hamburger or chicken or whatever, and you also have a salad, make sure that you keep those very separate because the one that won't be cooked at all is going to be particularly dangerous if it is contaminated. Okay, HACCP. This is important. Okay, so HACCP stands for Hazard Analysis and Critical Control Point. You need to know that. It is a food safety management system that helps identify and control potential hazards. And so rather than acting reactively to a food safety problem, like, oh no, we got these people sick, what do we do? It is a proactive management system where you are identifying where in your process potential hazards can be introduced and controlling those points in the process. So how HACCP was developed, it started in the 1960s. NASA worked with Pillsbury to develop a preventive approach to ensure safety during space flights. Because it can, as you can imagine, someone getting a foodborne illness in space would be really, really bad. So they're very, very strict standards, you know, a zero tolerance approach for food safety. In 1996, the USDA implemented HACCP and required it for all meat and poultry establishments. It took a few years for it to go into effect for everything, but this is when the rule was introduced that HACCP had to be implemented in all meat and poultry establishments. Okay, it wasn't until 15 years later that FDA mandated HACCP. So they were a little bit behind. So the HACCP requires 7 principles, and you could almost call these steps, okay? The first is conduct a hazard analysis. Now, before I continue, I want to say there's a list that's in a blue box in the book, and it has these in the correct order. But as you're reading the book, in paragraph form where it describes these principles, they're not in the correct order. And actually one of them is completely incorrect and describes the principle completely wrong. So rely on this that I'm giving you today. So conduct a hazard analysis. So this is looking at your flow chart that you've already made in your product development projects. And at each step, what are the potential chemical, biological, and physical hazards that might be introduced at that step? And also, how likely are they to occur? Is it something that could easily happen at that step? Or, well, yeah, I mean, there's like this slight chance, but really it's not an issue, okay? If yes, it is reasonably likely to occur at that step and it is a hazard, can we control it? And where is the point in the process that we control them? And so that is the second step, because you might control it perhaps somewhere at a different step, okay? So we're identifying the critical control points. Exactly where in this step are we able to control this hazard, this metal getting into the food, this potential pathogen that could affect the food, etc. Then we establish critical limits. So these are the measurable limits of this control that we want to ensure. So what is the maximum temperature that refrigeration unit can get to? What is the maximum time that a refrigerated product can remain in a warm loading dock before you have to take some sort of corrective action and to keep that food safe, okay? Fourth is establishing monitoring procedures. So you have to have a plan for how you're going to monitor these things. How often? How long? Who does it? Fifth is establishing corrective action. So what if you have a deviation? Okay, what is the corrective action you take? Can you recook the product to make it safe? Do you have to dispose of it? What is the thing that you do to make sure that only safe product is entering the food supply? Number 6 is establish verification procedures. And so for this one, you must verify that your process is working. Now, this is the one that is written incorrectly in the book. This is about verifying within yourself that your process is working. So you're checking the accuracy of your thermometers. Okay, if your thermometers are inaccurate, then the whole system is not working. You're reviewing records to ensure that your plan is being followed. That is verifying that your process is working. And the final HACCP principle is to establish record keeping and documentation procedures. You must have a plan for what records and documentation will be kept doing your HACCP plan and how those records will be stored. If you can't prove it, it didn't happen. And that is the way regulation works. FYI, so yeah, never mind. I already told you that the book is wrong on that one. On number 6. Okay. So last slide. This is a long lecture, but okay. So last slide. Government regulations. I've condensed what is in this section of the book, and you really just need to know what I'm telling you here. Okay. So meat and poultry slaughter production facilities. So we know that they are governed by, or regulated by USDA. In these facilities, a USDA, so USDA inspectors are required if the product is going to cross state lines. State inspectors are for if the product will always only sold within the state. Okay, so like the Texas A&M Meat Lab, they only have a state inspector because they don't sell things across state lines and they don't need a USDA inspector, or at least it was like that when I was a grad student. But these facilities, an inspector must be in the facility for any processing to occur. They can't do any work without an inspector there and able to watch them at any given moment. In FDA regulated facilities, the situation is quite different. FDA conducts routine inspections, and this is the actual regulation, at least once every 3 years for high-risk facilities and once every 5 years for non-high-risk facilities. So, now yes, I would say there's a lot more FDA foods that are lower risk than meat and poultry operations, and we've gone into why those foods are higher risk. But the difference in inspection would be laughable if it wasn't so scary to me. So there's a lot less food inspection in other industries than there is in the meat industry. Okay, food service facilities. So now here we're talking about your local deli, your local restaurant, etc. Okay, so these are going to be local health inspectors, and the terms of those inspections are going to vary from state to state, exactly what their regulations are, what they're looking for, etc. But in general, these facilities are closed if the score are low. I'll also mention, I forgot, like up here. So for both of these, the FDA or the USDA, they can force recalls of product based on their findings during inspection or reported incidences from consumers or the company. Now, the FDA one, it says at least once. So like if there are things being reported, they'll be coming in more often. But the minimum requirements are they're a little bit crazy to me. Okay, so last thing that I want to mention. I know that this chapter is probably a little bit overwhelming because there are all of these crazy names of organisms, and I've given you all these lists with times and temperatures and all this crazy stuff. So I'm going to kind of condense what is the most important when we're talking specifically about the organisms. Like, what do you need to know specifically about each organism? Okay. So you should know whether it's if for the bacterial or for all of them, whether it's a source of foodborne infection or it's a foodborne intoxication. Okay. You should know for each organism whether it is a bacteria, a virus, or a parasite. Okay. You should know the main sources of that organism, the main ways that it contaminates food, and which foods are particularly dangerous for each organism. Okay. And the last thing that I would suggest you really understand for each one is, well, not all of them. So I pointed out something special. If I've pointed out something special about the organism that makes it different or more dangerous in some situations, you should know that. So for example, Staph aureus is salt tolerant. Listeria monocytogenes is salt tolerant, and it grows at refrigeration temperatures. Okay, we talked about that, and it causes miscarriages, right? Like I like really tried to hammer that home. Clostridium botulinum, it causes that infant botulism, and so it's really important to not feed infants honey. Okay, so those kinds of things where I've pointed out like, oh, this is important, and here's why this bacteria is so important for human health. Pay attention to when I pointed those things out, because those would be good things to know. Okay, but like how many people get sick every year? I'm not going to ask you that, right? Exactly how many hours it is that it until from ingestion to infection or intoxication. I'm not going to ask you for those specific numbers. Might be good to know, like Staph aureus is like fast one, right? But other, I'm not going to, you don't need to know that salmonella is a 6 to 48 hour incubation time, for example. Okay, so don't so much focus on the little details, but focus on the overall, like, okay, this is within this group, or these groups, and this is the big thing that stands out about this one that Mrs. Cruzen pointed out. Okay. And so that is it for this chapter. Your food product development next project is going to be doing the first part of a HACCP plan. I'm not having you do a whole HACCP plan for this class, but it'll be the first part of the HACCP plan. So that's one of the reasons to really understand the HACCP principles. Also, okay, here, you will need to know these. Okay, it's going to be a 7-point question. 7 principles, 7 points in order. Okay, so start working on studying those now. All right. Okay, so I will see you in class.